Well, thanks. Thanks for the organizers for inviting me to this very nice meeting. I'm really looking forward to it, and also to letting me talk in the first day so I can have more beers in Sofia. So today I'm going to talk to you. So my lab is working mostly on functional organization of genomes in a variety of species, mostly microorganisms. But today I'm going to talk about a long-lived project in the lab that is mostly contextualized through evolution, on the fact that during evolution, species exchange DNA all the time. So this is a major driver of evolution. It helps genomes to complexify and to gain new functions. So these are some of the mechanisms that can result in the exchange of DNA between species. So for instance, horizontal gene transfer, when DNA exchange between one species to another, either prokaryotes on prokaryotes or prokaryotes on eukaryotes. You can also have viral invasions, transposons, waves of transpositions. For instance, you can also have introgressions when large pieces of chromosomes are exchanged between the two different species. So all these events combine to make genome more complex over time. And of course, there are also lab approaches where you can just inject DNA from one species into another. That happens all the time. And now with the field of synthetic genomics, synthetic biology, people are injecting large pieces of DNA all the time in genomes of different species. And so the common feature between all these events is the fact that you inject one piece of DNA into another species. And there is an immediate consequence, is that this DNA molecule is going to have to play by the host metabolic rules. So these rules, as you all know, are composed mostly of ingredients. So the DNA molecule, DNA binding proteins, physical parameters such as supercoiling, etc., that can combine to fold the DNA molecule into functional or not domains or loops or whatever. And the big question is, of course, does this interplay with functions? So either transcription regulation, repair, etc. So we will hear a lot about that in the next 3 days. And the main point is how to decipher these causal relationships between ingredients, folding, and functions. So when you inject a new DNA molecule that has not involved in the host forever, you basically don't play with, you don't do reverse genetics or anything like that, but you just modify the substrate of all the rules that are going to apply on this molecule. And so this features has been used in many studies to cast the rules of the host under a different light. For instance, Robin Koszul introduced chromosomes of Schizoscaromyces pombe into mouse cells as early as 1997, etc. And so this has been instrumental to learn a lot about metabolic function, of hosts. And so, since we work a bit on infection and these kind of things, I was wondering, can we play a bit with this question? And can we introduce and investigate how large, very divergent DNA molecules behave in a completely new, different context? And to do that, we turn to strains that were primarily developed by Craig Venter's lab, including by Carole Lartigue, who visited Pasteur almost 10 years ago and showed what she did during her postdoc. And I was intrigued by the fact that they cloned large bacterial chromosomes into yeast genome. So after discussing with Carole, we recovered the strains, and we started to linearize the bacterial chromosomes and add a yeast subtelomere, so they also have one yeast centromere. So now we have yeast strains that have the 16 native eukaryotic chromosomes plus one artificial megabase long stable chromosome coming from a bacterial species. So these bacterial species are either Mycoplasma pneumoniae, which is 40% GC rich, which is approximately the GC content of the yeast genome on average, 38%. Here you have the distribution of the GC block of 1 kb. So you can see it's centered around 30-40% for yeast on the Mycoplasma pneumoniae. And you have a native rich genome of 1.2 megabase called Mycoplasma mycoides, which is a different species. So now we have two yeast strains, one with Mycoplasma pneumoniae and one with Mycoplasma pneumoides extra chromosome. And we started to characterize the chromatin organization of these bacterial genomes. And so here I will define chromatin as a combination of nucleosome plus cohesins plus RNA polymerase. So we look at the three players using different techniques. So nucleosome positioning, this is done using MNAs, and you can see here in gray, it's always going to be the yeast, native yeast chromosomes in the strain. These strains are pretty cool because you have an internal control, so you can look at the yeast chromosomes and compare them to the bacterial chromosomes to see if they behave the same. And so you have the gray correspond to the MNase digestion of MNase-seq data of yeast chromosomes. And in purple, you have the Bordeaux, you have the pneumonia genome. So it's we can show, I mean, there are more data than that, but we can show that the nucleosomes are actually well organized and well formed along this DNA molecule. It's not true for Mycoplasma mycoides, so we also have more data showing that the nucleosomes are actually there, but the DNA is digested faster because it's AT rich. So the chromatin is not as clean in the MNase experiment. In RNA, in the MNase experiment. In RNA pol2 data, we can show that there is a lot of RNA pol2 along the M pneumo genome, but not as much along the Mycoplasma mycoides AT-rich chromosome. And when we look at cohesin, you can see here for two yeast chromosomes, the cohesin deposition with enrichment at centromeres, which is expected during G2. So in yeast, chromosomes are folded into chromatin loops during G2M stage, mediated by cohesin through a loop extrusion-like mechanism. So the loops are, I will show you later, but they are formed along these yeast chromosomes. There are also cohesin organization along the M pneumo with enrichment at centromere. And you can see here that on the M myco genome, which is AT-rich, you don't see any peak at centromere, and you actually deplete the cohesin from the centromeres of the native yeast chromosomes, but you can see actually an enrichment of cohesin along this bacterial genome. So this brief overview shows you that the regular GC content M pneumo genome behaves a bit similarly to the native yeast chromatin, and the AT-rich Mycoplasma mycoides genome is quite different from the native yeast chromatin. So we wondered whether or not we could learn to predict these features. So this was done with Julia Mosico. Okay, so this slide was supposed to be later, but this is transcription. So we also looked at transcription. And what we can see, it's quite fun, it's just that on the left here, you have the yeast genome sequence, RNA-seq stranded reads. So you can see in red the forward reads and in blue the reverse reads that follow, of course, the orientation of the genes along the yeast genome. On the pneumo genome, you can also see long tracks of transcription that actually follow somehow the orientation of the bacterial genes along the bacterial DNA, which was a bit surprising on average. On M myco, you don't see hardly any transcription, but still when there is transcription, the RNA reads tend to be on the same strand as the orientation of the bacterial genes. So there are something taking place here that makes that in yeast, bacterial genomes are still transcribed, at least the visible RNA that you recover follows the orientation of the bacterial genes. Anyway, so we wanted to see whether or not we could predict the chromatin features of this bacterial genome. So to do that, we worked with Julia Mosiconacci, a colleague in Paris, and his PhD student, Etienne Routier. And so we trained a neural network on the 15 yeast chromosome sequences, so the 15 first chromosome sequence of yeast. The deposition profile of either MNase, cohesin or NRE-pol2 or the MNase-seq data. And so they trained a neural network with this information and tried to see whether or not this trained network could actually predict the behavior and the deposition patterns of chromosome yeast 16, for which we have all the data, on the two bacterial genomes. And it works pretty well. So this is the MNase-seq data. So in gray, this is the yeast native chromosomes. In blue here, this is the prediction. And in gray, again here, this is the experimental data. So that's the average, and this is a zoom in on the results. So you can see that for MNAs, the nucleosome positioning is well predicted by the model, which was actually published by Julien on bioRxiv quite recently. On MICO, you can see that qualitatively you can keep the same prediction correlation, which is pretty good. On MPneumo, you also see that there is a quite good correlation between the prediction made by the network on the deposition, the MNS-seq experiment. It works also pretty well for RNA polymerase II. So this is native yeast, and this is the prediction by the network. You can see the peaks are well conserved. You can see a depletion in transcription along the MICO chromosome, which is predicted by the network. And now you can see also that the pneumo chromosome is actually quite well predicted by the network. And finally, for cohesin, it works also pretty well. So a neural network trained on a cohesin deposition profile is able to predict pretty accurately the deposition profile of other pieces of DNA in yeast. So you can see here on the MICO, you can see the enrichment I was talking about. So these are the experimental data, and these are the prediction. On the pneumo, you can see also that there are nicely well-defined peaks of cohesin deposition along this bacterial sequence. So overall, the trained neural networks are able to predict relatively accurately how native, but also artificial DNA sequences will chromatize with, including MNS-seq, POL2 and cohesin deposition data of given DNA sequences. So then we looked at the folding of these genomes. So we did a high-C experiment. I won't describe the technique, of course. And these are just the chromosome 15, chromosome 16 on the Mycoplasma pneumoniae genome of the high-C map. And so these strains are synchronized in G1. So you can do that quite easily using in-budding yeast. And this is a strain containing the Mycoplasma pneumoniae genome, and this is a strain containing the Mycoplasma mycoides genome. And you can see clearly that there is striking differences between these two maps. So this is a contact frequency according to the genomic distance map plotted here. And you can see that when you do that for the MPneumo strain, the MPneumo chromosome behave very similarly to native budding yeast chromosomes. So you cannot distinguish it clearly. But then if you go to the Mycoplasma mycoides strain, where the chromosome of MMyco is not really transcribed at this stage, you can see now that the slope of this contact frequency curve is closer to -1, whereas the other native yeast chromosomes are still behaving as expected. And so this suggests that what you can see also is on the map, you can see clearly that the extremities of this, the entire Mycoplasma mycoides genome seems to make more contact with the subtelomeres of the other chromosomes. So you can see here the stripes, the black dotted lines here, with the enrichment in contact all over in between the subtelomeric regions of these different chromosomes on the entire Mycoplasma genome. There is also a tendency for the Mycoplasma mycoides genome to make more contact with itself than with the rest of the chromosomal set. So all this information suggests that the Mycoplasma mycoides chromosome is quite isolated and is making more contact with subtelomeric regions of budding yeast chromosomes, which are typically positioned next to the nuclear envelope. And so the slope also suggests that this chromosome could be more in a globular organization than the rest of the chromosomes. And so we tested that with help of Angela Taddy and Myriam Ruot at Institut Curie, who did fish. So here the fish experiment was quite nice because we can just take the genomic DNA of the bacterial strain, the real bacterial strain, right? Label the DNA and then do fish on the yeast cells that contain this bacterial genome. So here the label is done with the entire mycoides genomes, which are labeled in green, and we also label the strain with DAPI to look at the rest of the chromosomal set. So these are the results for Mycoplasma pneumoniae. These are the DAPI, the Mycoides genome, and now the combination shows you that the Mycoides pneumoniae chromosome seems to is quite dispersed into the nuclear space. I don't know if it's the first time, but we can see, what we can see here is one chromosome among 17 chromosomes, right? So we can see that it seems to make little blobs in the nuclear space. We don't really know what they correspond to, but it's quite, it was quite nice to see that. When we do that for the Mycoplasma mycoides chromosome, which is inactive, now we have, instead of having this quite dispersed dot, we have, as you can see, one large bright dot in most cells, demonstrating that actually, yes, this chromosome, which in high C behaves like being individualized and quite insulated from the rest of the chromosomes, is indeed in this position, like make a kind of globule at the edge of the nuclear space. So this inactive additive-rich molecule is set aside from the rest of the active sequences in G1. So now we turn to G2. So as I mentioned, the G2 M cells in yeast are folded into coenzyme-mediated loops, similar to the one you find along human interface chromosomes. And so here you have the genome, the chromosomes 15 and 16 from budding yeast, and you have Mycoplasma pneumoniae chromosomes. So you can see that they behave quite similarly, and indeed, when you plot the curve, you don't see any difference between native yeast chromosomes and the Mycoplasma pneumoniae genome. So this is G1, and when you move into G2M, you have this arch, the shoulder in the curve, and that tells you that indeed the loops seems to be formed along the, are formed along the chromosomes. When you look at the Mycoides chromosome, which was quite different in G1, in the G2M, after replication, you can see now that the curve is actually identical for the Mycoplasma mycoides chromosome compared to the native yeast chromosomes. And indeed, in the map, you don't see any more differences between this genome, this chromosome, and the rest of the chromosome of the native yeast chromosome. So it means that the difference that existed in G1 is actually alleviated in G2M. The two genomes behave similarly, the bacterial genome and the native yeast genome. So it suggests that these AT-rich inactive chromosomes are actually folded correctly in G2M compared to G1. So we zoom in on this data. And what we can see here is the chromatin loops I was talking about. So along the Mycoplasma pneumoniae chromosome, you can see that it makes multiple loops and that correlate well with the coenzyme deposition sites. So there is discrete loops along the Mycoplasma pneumoniae genome, and the loop boundaries are associated with coenzyme peaks, which is actually expected on what is observed along the native yeast chromosomes. When we look at the inactive AT-rich sequence, it's quite different. So although the contact frequency curve fits with the native yeast curves, suggesting that the chromosome is well folded, it doesn't exhibit discrete loops. So now this is a piece of yeast chromosome from the same strain. So you can see the loops are not super visible, but this is not micro C, right? It's high C, but the loops are here, and they correlate well with the coenzyme deposition site. But then when you look at the bacterial sequence, you don't see any more discrete loops. But you can see that there is a huge enrichment in coenzyme that I mentioned earlier, and there is also a compaction of the genome, which is shown by the slope of the curve. So to demonstrate that this are actually there are loops actually mediated by coenzyme along this chromosome, we worked with a WAPL mutant. So WAPL is going to prevent the removal of coenzyme from the DNA. And in these conditions, in wild type, we know that it has been shown by multiple labs also in mammalian cells that WAPL mutants tend to make longer loops. So the curve is going to increase towards longer distances. So this is exactly what happens in WAPL. This is a plain line. The slope is, so not the slope, but the curve is moving toward the right. And for the pneumoniae chromosome, it's the same. and we observe exactly the same behavior for the Mycoplasma mycoides genome. So that suggests that the compaction we observe along this AT-rich inactive sequence is mediated by cohesin-mediated loop formation, except the loops are not discretely positioned. They are probably all over the DNA. And so to finish, we wonder whether or not we could play a bit with these sequences. So Léa Meneux, a PhD student in the lab, fused the Mycoplasma mycoides sequence with a native yeast chromosome. So she fused chromosome 16 with mycoides genome. So now we have one chromosome which has one arm which is native yeast and one arm which is mycoides sequence, DNA. And you can see here the high-C map, and this is a correlation map. So the DNA still is compartmentalized aside. Then we can introduce reciprocal translocations between the arms. So now we have chimeric arms with native yeast chromatin, mycoides chromatin, native yeast chromatin, etc. And we can also do reverse translocation. So now we can actually have strains with one chromosome which has pieces of bacterial and yeast DNA over various sizes. And you can see that you start to transform the map with this grid pattern that we are familiar with larger, more complex genomes. And what she did in the end was to translocate on another chromosome one of this piece of DNA. And now we have two chromosomes that carries each bacterial molecules along the bacterial DNA along their length. And now you end up with this very striking pattern that is really reminiscent of A-B compartmentalization of other species. So we don't have the fish data, but we strongly think that this shows that all these inactive DNA sequences is actually pushed aside from the active DNA sequence and still make this globular structure at the edge of the chromosomal pack. So compartmentalization is maintained independently of the dispersion of the DNA over the genome. So to conclude, we have two kinds of DNA sequences. So when we introduce a sequence which is at GC level more or less similar to the native GC level content, it is spontaneously treated, and we can predict it using neural networks like a native host chromatin, and it's going to be active, transcribed, etc., etc. So we did a mass spec analysis. There are not many proteins, but still it's quite intriguing. So it is actually treated as okay, I forgot. I'm missing half of the. Sorry for that. So that's not the. Okay. Sorry. So and then if we introduce a DNA molecule which is AT-rich, and it's not going to be treated like the host chrome. I mean, it's going to be actually treated differently, and it won't be active. You won't have active transcription, and the molecule is going to be set aside from the active DNA chromosomes. In G2M, you lose all these differences. The DNA is processed the same whether you are active or not. And this is also quite reminiscent with what has been found in mammalian cells, where A and B compartment disappear during metaphase and then reappear afterwards. So we have many more data, and so there is this preprint online if you are interested in. Léa has a poster on the compartmentalization. And with that, I will thank Christophe Chapard, a postdoc who did part of the experimental work, Léa Meneux, who did all the translocation analysis on the experiments, and Jacques Cerizet, who did all the analysis of the work, which was a very nice work. It's a strongly collaborative work. It's been in my lab over many years. Frédéric Becquet gave us some help with cohesin information. Julien for the neural network analysis, Carole Lartigue for the original strains, and Angela and Miriam for the fish analysis. I will also just briefly mention, I forgot to add the slide, but Amaury Bignot has a poster where we actually have a full genome sequence of bacteria which is turned off, and we have one single transcription track left on. So if you are interested in transcription, on high C, I suggest you go see him. Thank you for your attention.
Hi, very, very interesting talk. I was wondering, do you think it's the transcription per se that, the lack of transcription that forms this compartment, or the fact that they don't actually assemble nucleosomes? And have you tried putting an active transcription unit from the body or from the yeast genome into the bacterial chromosome, or like maybe an array of 601s or something to get back nucleosomes to restore the compartment?
Yes, of course. Yeah, no. So that's very good questions. So we have many data. I mean, more data suggesting that the nucleosomes on the AT-rich sequences are there. I mean, their histone chip shows you that there are histones. We have an MNase kinetic that tells you that there are actually well-formed nucleosomes along the sequence. It's just that it's digested faster because it's AT-rich by the MNase. So on the MNase experiment I show here, you don't see it very clearly. But so we think the nucleosomes are here. And so initially we wonder whether or not it's because the nucleosomes were not well formed, that they may stick together and you just create this bundle. But now we think it's maybe because it's actually quite inactive. So I think we still have to discriminate between the two, but I think it's actually active transcription that, inactive, the parameters. Then we also, the original idea with this construct was to do some bottom-up engineering of chromatin folding by adding active, inducible genes and so on. So we did that, and we don't observe. We observe many things, but not exactly what we were expecting. So we observe transcription-mediated loops, but independent from cohesin. So this is part of another work, but we have actually strong evidences for transcription-mediated loops on these chromosome sequences. And and so now the question is, we also wonder whether or not having an active gene in this compartment is going to affect its regulation, and these are experiments we are doing now at the moment.
The emergence of compartmentalization yeast is really striking. But my question is about the kind of compaction in G1. Does it decompact in G2 as it gets coated with cohesin, and especially in the case of Whopple, does it extend? That's the first question. The second one, is this compaction something natural that happens to naked or kind of AT-rich DNA? So if you just microinject plasmids, do they kind of clump together? Or is there some specific mechanism, like do they get coated with SIR proteins? Do they kind of get actively silenced?
Yeah. So we don't know. I don't know. We are trying to work on the molecules that would be inactive but not display the same AT composition, and to see if we can discriminate between activity, compaction, and compartmentalization. But
and in G2, so do they get, when they get coated with cohesin?
No, so in G2, we really think that the cohesins are loaded on this DNA, even though there is no transcription. So DNA cohesins are loaded, are making loops. But since there is no transcription to position the boundaries of the loops, to anchor the loops, then it's going to be different in every cell.
I meant to say just in imaging, do they get more decompact?
So we didn't do the fish in G2M, but we could do it. But yeah, we could do fish actually with single chromosome seeds. Yeah, that could be fun.
Okay. One quick, the high-C map you obtained for the Mycoplasma genome in yeast, did you compare it with the high-C map you would obtain if you come from bacteria?
Yeah. Yes, we have some data, but it's a very short genome, and so it's 1 megabase. And so, I mean, in bacteria, mostly what we see, I don't think we have a good high-quality maps for this species. So we had data, but like from 5 years ago, and the quality is not good enough to compare it with this. Because this is 1 kb resolution, and for the bacterial maps, I think it's like 5 kb. So it's not.
And second question about evolution, because I feel here you insert the genome and you look at how it is. For the pneumoniae, it's like, okay, you already have the GC content, which is according to, and for the microides, then it's 24%. It's very different, very alike the inactive regions in yeast. Now, if you let the yeast with the inserted genome evolve, having a lot of divisions, do you see transformation of the GC content in the bacterial genome according to then maybe starting cohesin transcription by eukaryotic pol 2, or I don't know if you...
Yes. So we put the strains in bioreactors also for fun, to play a bit with that, to see if we could actually, I mean, if the strains would evolve, but there is no selective pressure really on the strains are quite stable, the chromosomes are quite stable, so we see that they don't, we put the one with multiple translocations, so we see that the DNA is still there after 200 generations, which is not that much, but so it seems to be relatively stable. And the thing is, there is no strong selection pressure, so it's difficult to, I don't know how long we would have to evolve it to see the GC content to start to evolve in a certain way.
If you say no selective pressure, but if the genome starts to have mutations and yeast with more and more GC, then the polymerase would come on, the eukaryotic machinery would go on it, and maybe starting to have an evolution. And maybe then mixes, recombinations between eukaryotic and bacterial genome in this case.
We wanted for the yeast to adopt the ORFs present on the bacterial sequence, because there is a ton of information that yeast has never seen. But so far we haven't been able to, to do it.
Okay, so a very nice talk, and a striking phenomenon between the AT and GC regions. Have you looked on like a smaller scale at which point does this phenomenon like break down?
Sorry,
like at which point does this break down where maybe there's some stretches of like 50 kb or 100 kb or 10 kb?
For the compartmentalization?
Indeed, yeah. Where AT-rich regions don't really do this anymore.
Yeah, so 50, 45 kb, it's still compartmentalized. And we have 15 kb, and I think Leah was doing 5 kb. But we don't, under high C, you don't see it anymore. But we have to confirm it by fish to be sure that.
So it's based on the resolution of high C, basically. If you
maybe not. I don't know if it's. We don't see it on the high C, but it doesn't mean it's not there. Yeah.
Last question.
Okay, so quick question. I'm puzzled by the observation that transcription seems to be mostly in the same direction.
Yeah, but that's probably because there are regulatory pathways to degrade RNA molecules that contains stop codons too close to the ATG, so with a long tail. So I forgot the name of the pathway, but and so since the ORFs in bacteria, if you start to transcribe in the reverse way, you will hit these stop codons more frequently as if you transcribe in the forward way. It makes sense also that this is maybe one of the reasons. So we are testing the pathways to see if this is a reason. We don't think there is a specific determinant of transcription orientation in the DNA. I think it's just downstream regulation.
Okay. Thanks.
Okay, thank you again for the talk.